In the world of genetics, DNA is like a blueprint for who we are. It contains all the information that makes us unique, from the color of our eyes to our height and even our susceptibility to certain diseases. Each person's DNA is made up of millions of tiny units called nucleotides, which are arranged in a specific sequence. This sequence is unique to each individual, just like a fingerprint. When we are born, we inherit half of our DNA from our mother and half from our father. This means that we share a lot of our DNA with our parents, siblings, and other relatives. But how do we know how much DNA we share with a particular relative? This is where DNA matching comes in. DNA matching uses a process called genetic testing to compare the DNA of two people and look for areas where the sequences are the same. These areas are called DNA segments. The longer the DNA segment, the more closely related the two people are. But just knowing the length of the segment is not enough. We also need to know where the segment is located in the DNA. This is where a unit of measurement, called a centimorgan, comes in. A centimorgan is a unit of genetic linkage that reflects the probability that a pair of genes will be inherited together. The more centimorgans two people share, the more closely related they are. So, when we look at the length, location, and number of centimorgans of the DNA segments that the two people share, we can get a good idea of how closely related they are. This can be useful for things like family history research and medical genetics. That's the basis of how DNA matching works. It's a fascinating field that continues to unlock new secrets about our genetic makeup and our relationships to each other.